So good afternoon, and first of all, many thanks for the invitation today. It's a great pleasure to show you some of the work we do at SSI. And so my name is Madeleine, and very recently, I am heading the Metabolomics research group at the Danish Center for Neonatal Screening. And so my talk today will not be so much about specific research results, but more about our group's vision and the big potential I see in population size metabolomics methods to fundamentally change our understanding of health and disease. And so our group's main study objects are neonatal heel prick samples, which are routinely collected during the nationwide newborn screening. And all of those samples are then stored at the National Biobank, which I'm sure Lasse will tell you more about, is one of the biggest and most advanced biobanks in the world. And so currently there are over 2 million of those neonatal heel prick samples in the biobank, which corresponds to about half of the population of Denmark, and of course provides us with very extraordinary possibilities for population size metabolomics research. And so we can link those samples to diverse demographic and health-related data in the Danish registries. So our group's vision is that at some point we will have metabolomics methods that are good enough that we can predict individual disease risks and outcomes, giving rise to an improved understanding of disease risk, how we can predict it, but also how we can prevent it. And we think that metabolomics methods will be key to developing such predictive models. Why? So analogous to other omics sciences, metabolomics is defined as the measurement of all small molecules a human body synthesizes, metabolizes, ingests, or has come in contact with. And with that represents many important key factors that determine our health. And so, such as in the proteomics field, we use mass spectrometry-based methods to measure the metabolome. And the fragmented molecules provide us with a unique fingerprint that we can use to identify a chemical structure. For example, by screening to public data repositories, analogous to how you screen nucleotide or amino acid sequences. In big contrast to other omics sciences, however, where the chemical building blocks consists of a few amino acids or nucleotide sequences, the chemical space of the small molecules is vast. It's namely estimated that there is over 1 decillion possible carbon-based small molecules. And so this fact poses huge challenges to the chemical structural identification and biological interpretation of metabolomics data. And so this is also why metabolomics platforms, community-driven platforms, are extremely important. And so, sorry, I have a problem with my screen. And so one of those community-driven metabolomics platforms for data analysis and sharing is emerged in more recent years in the US, and it's called GNPS. And so GNPS provides researchers worldwide from diverse scientific areas with an interface where they can exchange and collect data on small molecules. So on the one hand, GNPS collects information on small molecules in a wiki-like fashion from the community, but also it provides different solutions to data analysis workflows that can enhance the chemical structural information we retrieve. And so I have worked with the group that has developed this platform at the University of California during my postdoc, and have also co-developed some of the computational workflows available therein. And while the platform is not yet optimized for the handling of person-sensitive clinical data, we already have applied some of the workflows available therein in-house to our data from the neonatal heel pricks with what I think are very promising results, some of which I'd like to share with you today. So in fact, there is yet very little known what shapes the neonatal metabolome and what metabolites we detect in the neonatal heel prick samples. And so for that reason, we assessed over 100 pregnancy exposures in a cohort of over 600 heel prick samples and their effect on the neonatal metabolome in collaboration with our colleagues at COPSAD. And as the time is very short, I will only show you some results as examples. So what we could observe, for example, is that there is an extremely strong trend based on the season of birth, meaning that babies born in summer differ metabolically very strongly from babies that are born in winter. And so we started to hypothesize why that could be and what metabolites would drive this strong seasonal trend. And so by using computational data analysis workflows and community data on the GNPS platform, we were able to see that eventually some of those metabolites are related to the seasonal consumption of fruits by the mothers during pregnancy. So as here exemplified by a bunch of molecules that are commonly found in citrus fruits and are mostly found in babies that are born during the colder season, where this type of fruits are typically also available in Danish supermarkets. Among the same lines, we found pesticide structural analogs, which are commonly found in fruits and vegetables, mainly in babies that are born during summer and autumn. And this could eventually be related also to the seasonal consumption of these fruits or vegetables by the mothers. And so this finding for us, this insight gives a very nice starting point, we think, to, for example, assess the relation of those exposure-related metabolites to seasonal disease outcomes. But I think it also shows very nicely why it is so important to share this data, as we would never have been able to identify those molecules were it not for a food or environmental chemist who shared his or her data in a public repository. And so in the same way, we also assessed all of the remaining 100 pregnancy exposures. And so what you can here see is each exposure represented as a colored dot and exposures that are related, connected through lines. And here all of the exposures that had a significant effect on the neonatal metabolome highlighted. So we could see that many exposures shape the neonatal metabolome, such as the proximity to rural areas, antibiotics use at birth, but also antibiotics use during pregnancy, the mode of delivery, or also other factors such as the mother's BMI and some maternal nutrition during pregnancy. So to us, this shows really that the metabolome has great potential to assess environmental exposures at high resolution, but also to assess these exposures in relation to diverse disease outcomes. We namely also found that the metabolome is predictive of diverse disease outcomes. So for example, based on the metabolome, we can predict the number of days a baby is hospitalized. So as a next step, we could eventually try to see which of those exposures have an adverse effect on the metabolome, and if we can define that, lead to a healthy metabolome, and which of those exposures have an adverse effect and eventually lead to disease. So just to summarize briefly, by using the computational workflows and data provided by the community on the GNPS platform, we were able to significantly increase the chemical structural information and subsequently the biological interpretation of our data. However, this was really only possible because we could make use of data that other researchers shared. And so we really think that in the metabolomics field, those sharing platforms will be key to advance our knowledge. Of course, we are in the clinical field, and data sharing is not always so straightforward. And also, of course, the question arises how much personal information is contained in the metabolome. And so to give you maybe a bit provocative example that I just like to leave in the room and maybe open for reflection before I close my talk. So we also assessed those diverse environmental exposures and their effect on the mother's blood metabolome. And what we could see is that we actually can get a quite surprisingly high insight into diverse lifestyle habits, such as the BMI, the age, diverse consumption of food products, alcohol consumption or smoking habits, proximity to rural areas, whether the person had pets or not, but also diverse social circumstances such as education and income. And so for that reason, I really believe that so the establishment of a specialized platform that can, in a secure manner, handle clinical metabolomics data, but also allow at the same time the fair data sharing with the wide metabolomics community will be key not only to advance the clinical metabolomics field, but also to advance personalized healthcare and diagnostics in general. And so with that, I'd like to thank all of our collaborators that make our work possible, especially everyone at COPSAD that's involved in a specific study, my group at SSI, all the funding agencies, and of course, to you, many thanks for your attention.
Thank you.